Next, we have Katie Atherton, who got her second dose yesterday, and so we have a recording of her playing, and she should be on the call listening, but here we go.
Hello, my name is Kathryn Atherton, and I'm a second-year PhD student in the bioinformatics program and a first-year urban trainee in Dr. Bhatnagar's lab. Today, I'll be talking about my summer project, which aims to answer the question, does racism drive urban soil microbial diversity? So first and foremost, why should we care about soil microbes anyway? Soils are a key metric of green space quality, and soils that host a diverse microbiome provide crucial ecosystem services to their local neighborhoods, such as providing nutrients to urban plants, sequestering carbon, and suppressing pathogens. Additionally, studies have shown that exposure to microbial diversity, especially during childhood, is correlated with a lower prevalence of inflammatory diseases such as asthma and allergies. Because of the important role that microbes play in human and ecosystem health, some people have argued that access to microbial diversity should be a human right. However, urbanization reduces microbial diversity and increases the abundance of human pathogens. Microbial diversity is driven by environmental factors such as green space size, impervious surface area, pollution, temperature, plant diversity and cover, and soil type. Racist policies may drive access to soil microbial diversity. For example, policies such as redlining have led to environmental factors being distributed inequitably across urban neighborhoods, typically to the detriment of racial and ethnic minority communities. However, the role of historical racism in structuring today's urban green space microbiome is unknown. I hypothesize that the legacies of redlining policies have depleted soil microbial diversity and increased pathogen loads in minority neighborhoods due to inequitable distribution of green space size, impervious surface area, pollution, temperature, tree canopy cover and diversity, and soil to the detriment of minority neighborhoods. To test my hypothesis, I have a stratified design to sample green spaces across Boston using the microbial DNA to measure diversity. On the left, you can see a map of Boston's racial and ethnic demographics. Darker colors mean that the neighborhood is a predominantly racial and ethnic minority community, and lighter colors mean that the neighborhood is predominantly white. On the right, you can see a map of the soil types of Boston. The red color shows an artificial landfill soil that was used to flatten out Boston, while the yellow is a native soil. For my study, I've chosen spaces in different green space types, on the different soil types, and in neighborhoods that are predominantly white, predominantly racial and ethnic minority, and somewhere in the middle. I'm very excited to thank the BU Initiative on Cities and the Biogeoscience program, and of course the Urban program, for funding this project. And thanks so much to my mentor, Dr. Bhatnagar, and my lab mates Nahuel, Corinne, and Zoe for their help.